Hi. Well, yeah, as you say, I'm actually a student at the University of Minnesota, but today I'm actually going to talk about things that I did during my master's at Western University with underreports at Faro. First, why would I study arid ecosystem? And that's kind of a question that I ask myself sometimes, especially because I'm from the rainforest, so I should really be studying this. But it's true, right? Arid ecosystem are really beautiful ecosystem, right? You go there, you see this beautiful landscape with plants that seem to survive there. But an important fact is that arid ecosystems have very different environmental gradient, right? They have high temperature, low moisture, and low nutrient. And you almost want to ask exactly how does those organisms can able to sustain these environmental changes and challenges. And more importantly, if you think about global warming as a problem that the society is facing, we kind of have worried about this kind of drought map that are showing that drought will not only increase as global warming increases, but also this ecosystem become very vulnerable and are really at a critical level. So I'm a mycologist. I do study fungi, so I kind of want to understand then what fungal communities are dominant in this ecosystem. And one in particular that I study are dark septate endophytes, and as the name says, those are dark septates, so they're dark, due to melanin in the cell wall, which make them resistant to UV radiation per se, and they have been studied to be pretty good colonizer of plant roots, especially grass in this ecosystem where they seem to be very abundant. So that being said, then there's two main systems that I kind of want to focus on. First is the grass rhizosphere, right, or those grasses that live in this semi-arid ecosystem, but also of what's known as biological soil crust. And this is a mixture of soil particle and microbial component, bacteria, fungi, plus a photosynthetic organism, which can be, in this case, moss, it can be cyanobacteria, or it can actually be lichen. And really, all together, they consider as the skin of the desert because they really mediate a lot of nutrient cycle as well as exchange. So we wanted to understand really what fungal communities are constituting those different components, focusing on dark septate endophytes. So the objective of this research was first to study diversity, so really what's out there, how they vary seasonally. So we're going to take different points throughout the year, and really what do they do in terms of plants? Do they promote growth or do they affect the plant wrongly by being a parasite or a pathogen? So my system suggested was in the Moab Desert. So we took different samples throughout the year from April 2013 to April 2014, really once a month. And then from there, we actually separate the data, the samples in half. One half was actually used for direct sequencing, so using Illumina on the iSeq platform that would tell us exactly what we have in terms of sequences. And the other part was used for culture, where actually we grow out the fungi, because that's the only way we can do manipulation, and we can actually have better identification of the fungi. So then we did seasonal variation, looking at them quantitatively throughout the year, but also looking at the growth promotion as well as the Xerophine Assay, which is really testing how well they can do in drought condition. And to kind of tell a little bit what I did, particularly we're sampling two grasses, Trophosis and Bromos, really in the rhizosphere soil. And then as far as biocross, we took all three biocross, so moss, lichen, and cyanobacteria, both on the surface and right below the surface. So we have the idea of above and below ground communities here. And I want to mention that throughout this talk, when I talk about above ground, I'm actually talking about surface of biocrust. But when I'm talking about below ground, really I'm talking about both the rhizosphere soil and really below the biocrust as well. So the first thing that we start doing is by just taking the first month and looking at how do the thing on top differ with the community on the bottom. And this is kind of what we found. So as you're going from surface to below surface, we already can see that the community were different in terms of their pigmentation. So we had fungi that were really darkly pigmented on the surface versus community that were actually lightly pigmented in the bottom. And that kind of made sense because you expect that things that are on the surface are more exposed to radiation per se. So you might want to have communities that are more resistant on the surface. So we can then do this for a lot of all the month, and then look at how does this changes throughout the year. I must say that everything on the surface kind of stayed the same throughout the year. Right? We only had these major communities of dark septate, but I was actually started looking at below ground. We can all see that, if you look at on the y-axis here, condensation rate, which is a term for like relative abundance, pretty much. As we go through the different months, we had this huge increase here in the summer, and then most of the year they kind of seem to have relatively the same abundance, which kind of tells us that these dark septate were increasing majorly in the summer, and that could be done because environmental factor or just because of the plant demand. But these were all cultures, so we weren't quite sure that this is the true story. So we wanted actually to see how the data look like on direct sequencing. So we can actually know what fungi these are and then kind of go back to the Illumina data set and see if that was the same thing happening. And luckily for us, as we look at kind of the same sequences now using Illumina, the direct sequencing, and then we can compare below ground, so below cross and rhizosphere. Again, you can see that in the summer months, so growing from kind of June to August here, we had this peak in both rhizosphere and below cross. Again, emphasizing that these actually were increasing relatively during those months. And that, again, we could refer it to environmental factors, especially solar radiation and temperature that were increasing during the summer. We didn't see strong signals for the rain. That's because in those areas you have monsoons events, so a big rain can actually change the community really fast. That might be why you actually see a peak around October there. But the general idea is that dark septate were increasing in the summer, and this increase can actually be linked to environmental factor. But part of us really thought that we could actually do a little more. So we wanted to actually first identify what fungi we have so we can actually compare them and do more interaction with plant per se. So here I'm really talking about like looking at things in the rhizosphere versus in the biocrust. And I'm going to probably go a little bit quickly through this before I get to the more important point. So literally, as we compare biocrust and rhizosphere, we saw a strong overlap in the communities, with the difference that actually they were just changing in terms of their proportion within the community. So I'm talking about two major groups. Plant-Fungal here, it's an order of fungal that's known to have a lot of dark septate endophyte. So you can see that for biological soil crust, you had a lot of them versus compared to the rhizosphere soil. And the Uratiales here are kind of known to actually have non-dark septate. Most of them would be lean. Again, you see kind of this shift from rhizosphere and biocrust soil. So now that we kind of had an idea of how they were varying and what they were, we could now ask the question on how exactly do they interact with the plant here. And what I did really was testing different plants with different fungi here, not only dark septate fungi that you have in the bottom here, but also like a bunch of different things to have an overall idea of the community. And we took measurement of stem growth, root growth, pretty much length, and we took also measurement of biomass, some of which I will show here. But to actually kind of give you an idea, first of all, we could see that there was, so if you see this dark septate, right, the one in dark, but a lot of things seem to promote growth of root, which was making sense for the system because if you think about desert plant grasses especially, right, they have those huge biomass in root versus shoot, which was impressive, but comparing it to the control, this is what we're seeing, right? You see almost like very few roots being made in the control versus a lot of roots being made, so really a root network being produced here. And more importantly, if you actually start looking at a lot of dark septate really closely and just looking at them in different microenvironments, we could also see that they were increasing really significantly stem length, and which actually this is what I'm talking about. I'm talking about stem length. You can really see that there's a difference between the controls here and things that were growing with dark septate endophyte, which then made us a strong case to actually say not only dark septate can be changing seasonally due to environmental factors, but it can also be changing due to the plant demand because the beginning of the summer is actually what the growing season of some of these grasses are. So it's because of the interaction with plants that they might also be increasing during the summer. Wanted to do one more thing, which was actually to test if these fungi can actually grow under low moisture conditions. And really here we did a xerophilic assay, which is having this media that has glycerol in it, so kind of reduce water potential. Then you can evaluate if the fungi that you have can literally do well on the drought or not. And so this is a scale that I had from no growth to actually total growth, which means covering the plate entirely. But here again, the point was that dark septate endophyte that I have were really growing pretty well in xerophilic conditions. So to kind of sum up some of this, the idea of this first part was that dark septate were, first of all, increasing in the summer, and that could be done to environmental factor or to the fact that they were beneficial to the plant in terms of helping the plant grow better by having longer roots and longer stem. But also more importantly, this dark septate could actually be a major factor in terms of growth of the plant in general, because that's kind of what they've been studying for. And we also think that some of this actually can almost be assimilated to mycorrhizal association, pretty much. So now that I kind of had all this knowledge, and which is kind of what my future direction is, I kind of wanted to ask, or I want to ask bigger questions, such as what exactly shapes microbial endophyte communities? And if I know those factors, are those factors actually directing the nature of the symbiosis? So are those factors telling us that these fungi will actually be more a mutualist or a parasite? And that's kind of, because in desert, we are all stressed, right? So it's a stressful environment, so you expect a selection with favor, more mutualist for the plant versus for the endophyte. So what I'm proposing to, on studying, which I haven't started done yet, is this. So in this hypothetical system, right, if you were to compare a plant that has different microbial communities in the leaves and in the roots, you can actually look at environmental factors, so either rainfall or temperature, and see how those factors changes the communities. You can also look at, given that in Minnesota, actually we have a lot of fragmentation of the prairie due to agriculture, so we actually have this strong interface between annual monocrop or gazebo crop and like native prairies. So then we can actually easily compare the communities between the two and see if there's any strong signals. And to do that, I'll actually establish different plots towards southern Minnesota, going actually from east to west here, and we suspect that this will be drier versus the east will be more wet, and we actually want to see how those fungal community will change using the same host, and if I can actually mimic similar data that I have previously, that the endophyte will actually be beneficial and increase plant growth overall. So, you know, if anybody has any input on that, I'll be happy to hear. So with that, I would like to thank both of my mentors, Dr. Poros Alfaro, as well as Mr. John May, who really have helped me shape this idea and really test these fungi, which people don't really like doing because it's a lot of plants that you're actually just testing every time. I also like to thank the YU Fungal Lab that has helped me doing this. Have the different professors who have helped me done with analysis and experimental design of this research. And this was done in collaboration with Los Alamos National Lab. They're the one who have the money to actually go collect every month and do implicant sequencing every month on like 7 different microenvironments. And then the DOE who provided money. And if I have time for questions, I'll take any question. Thank you.